Hi, I'm Sammy,
And I'm Grace.
and today we'll be talking about embryonic development.
When exploring the formation and development of an embryo, we explore the processes and stages of meiosis, mitosis, blastocyst, gastrulation, and body plan. To break this down, we'll explain each of them, beginning with meiosis. As you know, meiosis is the cell division resulting in 4 daughter haploid cells. Meiosis happens in order to make gametes such as eggs and sperm and involves crossing over for genetic variation. Meiosis begins with an egg having undergone DNA replication, so it has 2 copies of DNA, therefore double the amount of chromosomes. In meiosis 1, these copies divide evenly, making 2 separate cells, each with 2 chromosome pairs. Once sperm is present in the area of the cell, or the egg, the cells undergo meiosis 2, where the 2 cells split into 4 haploid cells, each with 1 chromosome pair. When the sperm enters the cell becomes a diploid cell. Anything that keeps an egg and sperm from joining would be considered a prezygotic barrier. A prezygotic barrier can be temporal, habitat, behavioral, mechanical, or gametic. Temporal having to do with different mating times, behavioral being different mating rituals or attraction methods. Mechanical meaning the organisms have morphological differences that prevent successful completion, and gametic meaning the sperm of the male species can't fertilize the egg of the female species. After meiosis, mitosis is used to multiply and produce exact copies of these diploid cells. The diploid cells are stem cells, meaning they have the ability to become any cell in the body. Stem cells are used throughout an organism's entire life for growth and repair. The blastocyst stage is when the cells form a hollow mass that consists of 3 parts: an outer trophoblast layer, an inner cell mass, and a fluid-filled cavity. The inner cell mass is the source of embryonic stem cells. Gastrulation is the process of forming the digestive tract. The process involves the reorganization of a single-layered blastula into a 3-layered structure. These layers are known as germ layers and are labeled as the ectoderm, mesoderm, and endoderm. In terms of the fate map, the ectoderm will end up as neural tissue and the epidermis. The mesoderm will develop into skeletal and muscle tissue, dermis or vertebrae, and the endoderm turns into the digestive and respiratory systems. Gastrulation differs for some animals, but is generally the same for most. For humans and other animals, it happens in a certain order. First, the embryo becomes asymmetric. Then the primitive streak forms. Finally, the cells from the part of the primitive streak form the germ layers. During a phase of gastrulation called invagination, the cells fold in, forming the blastopore. In some organisms, the development stops at the indent, meaning the organism only has one hole for their mouth and anus, such as an octopus. Otherwise, the indent continues to the other side of the cell to form a tube, one side as the mouth and the other as the anus, which is common for most animals. Depending on whether the mouth or anus is the one to form from the blastopore, an organism is either a protostome or a deuterostome. In protostomes, the mouth develops first, whereas with deuterostomes, the anus develops first. The next step in development of an animal is the embryo body plan. This includes the head, body, front and back, and the sides. The body plan genes and signaling pathways are similar for all animals. However, the formation of the body plan is where embryos begin to look different. If the execution of the body plan is disrupted, mutations such as body parts in the wrong areas may occur. For example, in the mutations of the Drosophila fly having legs where its antennas would usually be. In summary, a sperm fertilizes an egg, the cells multiply, then form a hollow structure, then proceed to form the parts of an animal. The growth of an embryo is fueled by the nutrients and blood provided from the mother.
Created using Powtoon.